Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. All the honor participants of the first international conference on animal research for eco-friendly livestock industry. First of all, I would like to thank you for a moderator for this parallel session. This time, before starting our presentation today, let me introduce myself. I'm Wahyu Ekasari from the Faculty of Veterinary Medicine, Syiah University, Banda, Aceh. In this opportunity, I will present our research entitled Isolation and Identification of Fungus in the Feces of Hutan Sumatra goats, Capricornis sumatraenis. And this is what our research has been looking at. So let's get started. Forest goats are relatively small ruminants belonging to the subfamily Caprinae, genus Capricornis, and are divided into 3 species including Capricornis sumatraenis. The Hutan Sumatra goat is a forest goat that lives in the tropical forest of the island of Sumatra, and the distribution of these forest goats is found in mountainous and highland areas. Hutan Sumatra goat are mammals that live solitary and away from human settlements. Forest goat conservation began with the enactment of Act on the Protection of Wild Animals in 1931. The International Union for Conservation of Nature and Natural Resources has listed the forest goat as 35 endangered species. Several factors cause a decline in the population of Hutan forest goats, including depletion of natural habitats, excessive cultivation, hunting, competition with humans in the use of food sources, and death due to predators or disease. Disease identification needs to be done to determine the health condition of wild animal in nature, and this is one of the efforts that can be done to protect the forest goat population. Examination of microbes in feces is one way to identify disease that can be done. One of the sample collection methods that can be carried out on animals without causing stressor and is safe to do is a non-invasive method. So, based on this background, it is necessary to research fungus identification in Hutan Sumatra goat feces. This research is a preliminary study that is part of a large study on Hutan Sumatra goats. This research was conducted from September to October 2021 in the microbiology laboratory of the Faculty of Veterinary Medicine, Kuala University. The samples used were fresh feces from 10 Hutan Sumatra goats. Then the first culture was carried out in peptone water containing mushrooms, was treated onto several dextrose agar by forming a quadrant streak, then incubated at room temperature and observed for 3 until 7 days. This research is presented in the form of images and analyzed descriptively. Okay, we are going to result and discussion. In this section, we can see that according to 10 samples of Hutan Sumatra goats, so that 4 out of 10 samples contain fungi with 2 different types of fungi as shown in figure 1. Based on observation on the 4 samples that showed fungal growth on SDA media, there were 2 types of fungi, Saccharomyces spp and Aspergillus spp. Samples number 1, 2, and 5 showed the formation of yellowish-white colonies located on the agar medium surface. The edges of the colonies were circular with a shiny surface and grew in clusters. So samples number 1, 2, and 5 were identified Saccharomyces spp, while samples number 4 was identified Aspergillus spp. The finding of Saccharomyces spp in the feces of Hutan Sumatra goats are still considered normal because this fungus is a normal flora found in the digestive tract of ruminants such as cattle, sheep, and goats. The previous study state that the Saccharomyces and Aspergillus could produce amylase, protease, lipase, cellulase enzymes, was also a good source of B vitamins. However, not only species of Aspergillus provide benefits to animals. Several species of Aspergillus can have a negative impact and cause aspergillosis in animals, such as Aspergillus fumigatus, Aspergillus niger, Aspergillus flavus, Aspergillus cereus, and Aspergillus nidolensis are opportunistic pathogens for animals and humans. So, the conclusion of this research, based on the study result, it can be concluded that from 10 samples of feces of Hutan Sumatra goats examined, 4 fecal samples were isolated by fungi and identified as 3 samples of Saccharomyces spp and 1 sample of Aspergillus spp. In addition, the suggestion from this research is the identification of Aspergillus species using molecular analysis. Finally, we thank the Laboratory of Microbiology, Faculty of Veterinary Medicine, Kuala University for the facility provided. Thank you very much for your great attention, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh.